Mitosis is an important player in the play of life. The regeneration of a cell is what keeps life alive. Mitosis divides cells into single nuclear division. From this division, two identical daughter cells are born. Mitosis is important for growth and repair, keeping all living organisms thriving. Meiosis is the other important player in this play. Without meiosis, this play would be virtually impossible. Meiosis results in the production of haploid cells. When two of those cells come together, one from the female and one from the male, it creates a diploid zygote. This process creates diversity and creates more organisms. Mitosis, process of asexual reproduction ending in a diploid cell, functions in cellular reproduction and general growth and repair of the body. Asexual reproduction occurs in all organisms and is genetically identical to its parent cell. Meiosis, cellular reproduction where the chromosomes are reduced by half through separation of homologous chromosomes creating two haploid cells, functions in sexual reproduction, occurs in humans, animals, plants, and fungi, and is genetically different than the parent cell.
Interphase begins with the G1 phase. G1 is the extended period of time where the cell grows and carries out protein synthesis and several other cellular functions. Now the cell only contains one molecule of DNA, compact chromatin that is yet to be replicated. Now we start the S phase where a cell begins to replicate its DNA. At the end of this stage, all of the chromosomes have two chromatids. Completing interphase is the G2 phase. Cell continues to carry out normal functions and continues to grow. Towards the end, cytoplasmic organelles replicate and prepare for the cell to enter mitotic phase. DNA begins to condense. The nucleoli disappears. Centrioles move to the poles of the cells. The mitotic spindle begins to form from the poles toward the equator. The nuclear envelope breaks down and the membrane is pinched away. Sister chromatids line up at the equator of the cell known as the metaphase plate. The polar fibers begin to form and attach to the centromeres of the sister chromatid pairs. The sister chromatids split and move towards opposite poles of the cells, separating it and now being called chromosomes. Half the chromosomes, half the sister chromatids, move toward their own pole with the help of the mitotic spindle. During telophase, daughter nuclei and nuclear envelopes start to reform and the chromosomes decondense or uncoil. During cytokinesis, a cleavage furrow begins to indent around the middle of the cell that pinches the cell and dividing it into two separate and genetically identical cells. Two genetically identical diploid somatic cells form. Each cell is the same as its parent cell, resulting in 46 chromosomes and maintains the same job as its parents had. There are now two diploid cells ready to follow the tracks of the parent cell.
Homologous chromosomes come together and match up in tetrad. The tetrads line up at the metaphase plate of the cell. The homologous chromosomes separate. One of each pair goes to the opposite pole of the cell, changing the chromosome number from 2N to N or diploid to haploid. The sister chromatids stay together. Two daughter cells are formed and immediately enter meiosis II to separate the sister chromatids.
The nuclear membrane and the nuclei break while the spindle apparatus begins to form and the chromosomes start to migrate. The chromosomes line up at the metaphase plate at the cell's equator. The kinetochores attach to the sister chromatids, preparing them for separation. The sister chromatids begin to move toward the opposite cell poles after being separated with the help from the spindle apparatus. In telophase, two nuclei forms at the opposite poles. In cytokinesis, the two cells are cleaved, creating four daughter cells. Four daughter cells are created, each with 1/2 the number of chromosomes of the original parent cell. All four cells now contain 23 chromosomes and are ready to meet with the opposite sex cell to create a diploid, 46 chromosomes, zygote. A mother's gamete cell, her egg, is now met by a father's gamete cell, his sperm, and it creates a zygote.